uh, identifying DNA as the genetic material. The scientists actually did a great work to be able to know that the genetic material was actually DNA. DNA stands for deoxyribonucleic acid. DNA stands for deoxyribonucleic acid. So this was actually identified by the scientists as a genetic material through a series of experiments. So we're going to look into some of these experiments today. Among these experiments is Grifts experiment. Grifts was one of the great scientists which actually did an experiment which is called the transforming principle. So what Grift did is he experimented with a bacteria that caused pneumonia. So pneumonia is known to be caused by a bacteria. So then what he did with this bacteria is he used two strains of these bacteria. So he used the S, which is known to be the deadly, and the R, which is known to be not the deadly one. And as you all know, that pneumonia is an acute respiratory infection, and which is caused by streptococcus, and also can be caused by some viruses. So pneumonia is not something which is caused by one single organism, but we know that streptococcus pneumonia is one of the bacteria which actually causes pneumonia. So it used two forms of it: S form, which is known to be a deadly form, and a R form, which is known to be not a deadly one. So what did it do with these two forms? So a transforming material uh, passed from the S bacteria to live R bacteria, making it to be deadly too. So take a look at these the pictures down here. Now the live S bacteria is being given to this mouse. Then the mouse was found dead. Then the live R bacteria, which is known to be not deadly, was given to this mouse. The mouse did not die. So, but when he killed the S bacteria, it killed it. So it noticed that the S bacteria, when after being heated, when it killed all the bacteria in it, the mouse became alive. It didn't die when it was being injected. But when he combined the heat-killed S bacteria with the live R bacteria and he injected the mouse was found dead. So this is quite strange. That how would it be that the live R bacteria did not kill the mouse, and when live S was being heated, it did not kill the mouse. So what made it to kill the mouse when it was being combined with a not deadly form? So this is what brought about the transforming principle that says there must be something which is in the heat-killed S bacteria, which is being transferred into the live bacteria that made it active to kill the mouse. So that was when the research continued to get more advanced. So they Avery also identified DNA as a transforming principle. So the experiment, which was done before, so it reached the conclusion that the DNA, which is the genetic material, was transformed, was taken or transferred from the S bacteria, which is known to be deadly. The fact that it was being heated and the bacteria were killed does not mean the genetic material cannot still be transferred. So this is what made it to be transferred to the live R bacteria and made it to be effective in killing the mouse. Avery identified DNA also as a transforming principle. Avery isolated and purified Grifts transforming principle when he did when after the Grifts experiment was concluded. performed 3 tests on the transforming principle. What it did is called a qualitative test, which showed DNA was present. It also did a chemical test, which showed that chemical makeup matched that of the DNA. I mean, the DNA is composed of several things, which we're going to talk about later. It's composed of ribose sugar, deoxyribose sugar. It's composed of, so it's composed of deoxyribose sugar. It's composed of phosphate, and it's also composed of the nucleotide bases. So when he did the chemical test, it found out that the chemical makeup matched that of the DNA. And also, he did another test called the enzyme test. It showed that only the DNA degrading enzyme stopped the transformation. So there was an enzyme which can degrade, which can destroy the DNA. So when he did the test, he noticed the transformation was stopped because the DNA had been deformed or had been transformed or been destroyed. So this is the result of his chemical analysis of the transforming principle. Now let's move on to the next, to Archie and Chase confirmation of the DNA as a genetic material. Archie and Chase studied viruses and infect bacteria. We have a specific virus that infects bacteria, which is called bacteriophage. Bacteriophages are known to infect, or they not to attack bacteria cells. So, but they actually viruses, they're being called bacteriophage because they infect bacteria. So they tag them, they were being tagged viral DNA with radioactive phosphorus. So now what happened is they tagged a viral protein with a radioactive sulfur. So the tagged DNA was found inside the bacteria. I mean, the tagged protein were not. So the DNA was found in the bacteria, as you can see, but the protein was not found in the bacteria. The meaning is when this virus infects the bacteria, what is sent or what it inserts into the bacteria is actually the DNA, not the protein. So this made the scientists to understand that it's not the protein which is actually responsible for the transformation process or as for the genetic material. It is the DNA, not the what, not the protein. So this is all we have for 8.1. Now let's proceed from 8.2, which is a structure of the DNA. After we've already known that DNA is a genetic material, the scientists want to know what is the structure, what structure does the DNA have? So DNA structure is the same in all organisms. There is no difference in it. And the composition of the DNA is the same in all organisms. DNA is composed of 4 types of nucleotides. The DNA is made up of a long chain of nucleotides. Nucleotide bases, they are the base pairs which we have that make up the DNA. The DNA is made up of this long chain. The meaning is the bases are arranged in line to form a long chain. Let's say, for example, we know the DNA, which is the deoxyribonucleic acid, is composed of 4 proteins, which is adenine, thymine, guanine, and cytosine. So this is the components of these are the nucleotide bases. So each nucleotide has 3 parts. One of these parts, the phosphate group, the deoxyribose sugar, and the nitrogen containing base. So these are the 3 components of every nucleotide. And remember, the nucleotide is known to be the is not to be the components of the DNA. So, but the DNA, the nucleotide is made up of 3 parts. And remember that we talked about the monomers and polymers in before. The monomers are the small molecules of a large molecule. DNA, or we say, you can say nucleic acid is a polymer, and nucleotides are the monomers of the nucleic acid. Nucleic acid are divided into two. We have deoxyribonucleic acid and we have the ribonucleic acid. So the nucleotide is a monomer of the ribonucleic acid, or let's say nucleic acid. So in which DNA is one of the types of the nucleic acids we have. So the nucleotide is now being made up of three parts, which is the phosphate group, the deoxyribose sugar, and the nitrogen-containing bases. So this is what we have here. So now the nitrogen-containing bases, they are only they have only differences in four nucleotides. So what does it mean by this? The meaning is some of these nucleotide bases, they have a double ring, while some of them have a single ring. As you can see, the thymine and cytosine, they made up of single ring, and the purine and the which is made up of the adenine and the guanine, they are made up of a double ring. And this is the reason why there is when there is a pairing between these four nucleotides, we have to have one single ring with one double ring, and we have one single with another double. If you have two singles pairing together or you have two doubles pairing together, you're not going to have a good result. The reason is because the size of the DNA molecule would not be the same throughout the pairing, because a section where you have two double rings pairing together will be larger in size. The weight will be the size will be bigger than when you have two single coming together. So this is the reason why the scientists say that in the pairing it has to be T with A and C with G. So Watson and Crick, they actually did a great work on trying to find the structure of the DNA. So they actually did a lot of work here, whereas we had some other scientists who did better work than them, like but some of them died before uh, they were actually given the award for the job they did. So these Watson and Crick realized that DNA is a double helix that is made up of sugar backbone, sugar phosphate, which is the backbone, and is on the outside of the bases. The bases are inside, so the bases are inside, while the sugar is on the outside. So Watson and Crick discovery built on the work of Rosalind Franklin and Erwin Chagall. These two scientists were great scientists. Franklin's X-ray images were the one that gave the suggestion to Watson and Crick on what the DNA looks like. So what happened is the DNA was double helix and is even in width. So the width was even because one double ring pairs with another single ring. So one pairing with one pyrimidine, one pairing with one pyrimidine. So Chagall rules state that A will go with T, while C will go with G. So now the nucleotide always pair in the same way. There is no time you see the pairing happening, it's going to pair in the same way. And how does this happen? A will pair with T, and C will pair with G. So this is the reason why there is we got even width of the DNA strand or DNA molecule, because a pyrimidine, which is a single ring, paired with a purine, which is a double ring, and the helix has a uniform width. This is exactly what we're working towards. Now, this is exactly what it looks like when the combination is completed. So the backbone is connected by covalent bonds, but the bases like the thymine, the adenine, guanine, cytosine, they're connected by weak hydrogen bonds, while the backbone is connected with a very strong covalent bond. So the bond between the bond which is between the guanine and the cytosine is not to be a very weak, what do you call it, weak hydrogen bond, but the one between the phosphate molecule and the ribose sugar is not to be a covalent bond, which is a stronger bond. And all this make up the backbone. What this is the basis, which is forming the inside of the DNA molecule. So this is all I have for 1 and 2. Thanks for listening.